Okay, bio students, the purpose of this screencast is to give you some notes on the cell membrane structure and function, and this may or may not be my fourth recording of this. What is the cell membrane? The cell membrane, it's the covering for all cells, meaning if you're a prokaryote or a eukaryote, you have a cell membrane. If you're a plant cell or an animal cell, it doesn't matter, you have a cell membrane. And you have it, right, in order to kind of hold your cells in, but also for transport. And so transport be one of the main functions. And transport is basically just that movement of materials. You might remember that we described the cell membrane as being semi-permeable. Okay, and that permeable word would mean that stuff can just kind of freely pass through. Semi-permeable means that not everything can. So we would say that some materials can pass easily through the membrane and others cannot. And so maybe you read the Transport Proteins on Strike play, and you might remember then that the phospholipids had a chant of some things can pass, other things cannot. And that's where that comes from, that idea that the cell membrane is semi-permeable. Okay. When we talk about transport, we have two categories. We have passive transport, which we'll talk about in a second. And we have active transport. Active transport, I'm going to tell you a couple things and then just kind of leave it at that for now. Active transport, it's called active because it requires ATP. ATP, right? Cellular energy. And frequently, it's the movement of materials from an area of low concentration to high concentration. And so we say that this is we describe it as being up the concentration gradient. So concentration gradient meaning difference in concentrations, up meaning you're going from low to high. And so this is going to require ATP, right? It's just like if you push a ball up a hill, you're going to need a lot of ATP or a lot of energy. And so active transport is going to require energy. Passive transport's different because no ATP is required. And you've already learned about one type, which is called diffusion. And diffusion, we say, is the passive transport of particles from an area, so it's moving materials from a high to a low concentration. Right? It's like when particles spread out. We say that this is down the concentration gradient. So you saw this when you see it all the time if you spray like an air freshener in the corner of a room and the particles spread out, right? They go from that area of high concentration where you sprayed the Febreze to an area of low concentration where there's no air freshener. We're going to talk about a couple different types of diffusion. So we have facilitated diffusion, which basically means if particles are moving from a high to a low concentration, but they need a transport protein. The transport protein will help facilitate this process. Sometimes students get confused and say that facilitated diffusion, because you need a protein, you need ATP, but that's not true. No ATP is required in this process. Okay? And it is also still movement from high to a low concentration. The next specific type of diffusion that we're going to talk about is osmosis. So osmosis is just the diffusion of water particles. And so water, right, molecule of life, So it gets a special name. When water is doing diffusion, it's called osmosis. The next part that we're going to talk about is things that can pass directly through the membrane and things that cannot. What this is basically saying is things that can pass directly through the membrane, we're talking about things that do not need a transport protein. In other words, they can do simple diffusion. Okay? Talk about that in a second. The next category that we're talking about, so we're asking about things that cannot pass directly through the membrane. What this means is we're talking about these need a transport protein in order for diffusion to happen, or really any movement, okay? And so these would be things that do facilitated diffusion. All right, so when we talk about things that can pass directly through the membrane, we're talking about things that are mostly small and non-polar, meaning they don't have that polarity like water does. And so that basically means that they can squeeze through the phospholipids, okay? And so when we talk about small and non-polar things, some good examples are carbon dioxide, which is helpful because that's a waste, right? It's helpful they can just pass through. And oxygen is another good example. We really need oxygen, right, for cell resp. I'm going to put water in the middle of this chart. And so water is small, but it is also polar. And so its size helps it gets through the phospholipids a little bit better, but its polarity makes that a little bit challenging, okay? And so when we talk about water, we say that it can pass through, but it moves very slowly through without a transport protein. It moves much quicker if it does have a transport protein. And its transport protein, just like osmosis has a special name, its transport protein also gets a special name. It is called an aquaporin. Other things that can't pass directly through the membrane are things that are large, so they can't squeeze through the phospholipids, okay? And so things that are large include like glucose and amino acids. Other things that struggle are charged particles like ions. And so they will need a transport protein. Okay, and that's because charged particles are definitely polar. So hopefully this makes sense. We're going to go through and we're going to label the structure of the cell membrane. So before we talk about the structure, let's remind ourselves that the cell membrane is made mostly of phospholipids. And so phospholipids look kind of like this little guy I drew here. And they have a top part, the part that has the phosphate, which we call its head. And we say that its head is polar, just like water. And polar things like other polar things. So we say that it is hydrophilic, which means it is water-loving. The bottom part, the tails, which are made of the lipids, or like fats, are nonpolar. And we know that fats don't like to mix with water. And just like that, our lipid tails are hydrophobic, meaning they are water-fearing. Okay. So what I want you to do is I would like you to pause the video and also add in H2O movement. You should draw an H2O movement. And to take a second and to try and label these different parts of this cell membrane. I'll remind you that outside of the cell, it's often a watery substance. And inside of the cell, we're talking about our cytoplasm, which is also watery and polar. All right, so go ahead, pause the video, label this on your own, and then I will tell you about what it should be. Okay, so let's label our general structures, and then we can talk about how things move. So when I look at this, I see my phospholipid bilayer is the two layers of phospholipids. And let's recall that because the inside and the outside of the cell are both watery environments, we have 2 layers of phospholipids so that the heads can be on both sides, right? So we have this. So we have our hydrophilic heads are on both sides of the membrane. Inside of the membrane, though, are our hydrophobic tails, or our nonpolar area. Hydrophobic tails. And then finally, these big purple guys, these are transport proteins. And so something that I want you to realize is that this diagram is kind of limited. There's only one transport protein shown, but let's remember that transport proteins are very specific. They are specific to whatever they let move through. And so if I want to be able to let 3 things move through my membrane that need transport proteins, I will need 3 different transport proteins, okay? So I will say something right here, and we'll start with this. Glucose needs a transport protein, and sodium needs a transport protein. They need separate and specific transport proteins. So just as a heads up, that's a limitation of this diagram. Okay, so when we talk about things that move through transport proteins, need them with certainty, we can draw glucose. It can't squeeze through our phospholipids, so we'll draw it going through our transport protein. Sodium also needs a transport protein. Let's label it like this, sodium ions, right? Sodium is a charged ion. So while it and glucose would not go through the same transport protein, we'll still draw it going through this transport protein because we only have one on the diagram. When we talk about water, so water is unique, right? It goes much faster when it has its own transport protein, known as an aquaporin, but it can go, maybe we'll draw these arrows kind of slow, right? It can squeeze through the phospholipids. So water, it can move slowly through phospholipids. But it moves much quicker when it has an aquaporin or its specific transport protein. Finally, the last 2 ones that we want to label are carbon dioxide, and we're looking for carbon dioxide and oxygen. And remember, CO2 and oxygen, they are both small and nonpolar. So they will both be able to just squeeze directly through the phospholipids. They squeeze directly through. Okay, so hopefully this makes sense for you. And you were able to label your diagram as such. The last thing I'm going to do is just really quickly try and do some bad highlighting, right? Just remembering that anywhere that is touching water is going to be hydrophilic. So all these phospholipid heads on both sides of the membrane. And anywhere that is not touching water, just like the center of the membrane, will be hydrophobic. So here's my final highlighting for you. Okay, let's hopefully this helps.